So while we've gained a lot of information from studying aging in both cell lines and model organisms in the lab, as well as studying patients who have age-related diseases like progeria, and by studying long-lived animal species in the wild, the future of sort of aging research is focused in what's known as systems biology. And there's some particular reasons for that and some particular things that make systems biology a really kind of attractive approach for studying aging. And so, what systems biology is basically just a way to mathematically model um, complex systems to predict outcomes. And so, systems biology uses relationships and connections that we already know exist within these complex systems to make these predictions. And so, this complicated diagram here on the left is basically a system of aging in C. elegans. And so this is just showing the genetic network and relationships between different genes, which are indicated by the nodes or the dots in C. elegans, and how they're connected to each other and how they interact, which is indicated by the lines, in order to kind of affect aging. And so you can see here that there are a lot of genes involved in aging and longevity, and that they're not just interacting necessarily with one other gene, but they can be interacting with multiple. And all of these interactions, if we had to study them in the lab individually, would be almost impossible to fathom. And that is why we have to use kind of the power of computing and math in order to start understanding these really complicated systems and answering complicated questions. And you can imagine that to make the mathematical models used for systems biology, you're going to need to generate all of this data in the first place. You need a shit ton of data in order to make the models that we use to study systems biology or in systems biology. And fortunately, we do actually have shit tons of data available out there. These data are available in data sets from what we commonly call those like omics fields. So genomics, proteomics, transcriptomics. The Human Genome Project has generated a ton of data, for example, outlining every single base and every single gene in the entire human genome. And all of that data is publicly available. And so systems biologists can go in and use these data to make the mathematical models to start studying different complicated systems. And the same is true for studying all of the proteins in an organism, or the proteomics, all of the RNAs, so transcriptomics, and even all of the epigenetic modifications of these genes in the DNA. So there's a field of epigenomics that have generated these data and that we can use to study aging. And the reason it's ideal for studying aging is because, as we have talked about in the previous lectures, aging is a really complex system. And if environmental factors play such an important role in aging, which we know that they do, and two individuals cannot really have the same set of environmental interactions throughout their life, the possibilities for what's affecting the rate of aging in an individual are like almost impossible to fathom in number. And only really like a computer or a mathematical modeling technique can start to tease apart the mechanisms and actually make some conclusions about the mechanisms behind aging, especially in regards to the environmental contributors. And so, in order to develop these predictions as well, systems biology not only needs a lot of data, but they also need what are known as biomarkers. And so, if we're interested in studying aging, these would be called age-related biomarkers. And what a biomarker is just something that is used as an indicator of biological state. And so this can be an observation, so something that you can just see from looking at a person, so maybe, eye color. It can be a substance or a molecule, so a protein or a chemical or metabolite in the blood. And whatever it is, it can be used to indicate what biological state that organism is in. And so for an age-related biomarker, there are several tenets that it would really need to fill in order to be useful in systems biology. So it would have to be able to predict the rate of aging. It has to be involved in something that underlies the cause of aging. So some sort of process involved in aging, the biomarker would have to be a part of. Also, importantly, biomarkers have to be able to be tested repeatedly in the same individual without causing harm. And so you can imagine that something that's an observation, like looking at maybe eye color or hair color or particular like muscle change in muscle mass, is something that you can test repeatedly without causing any harm. It can also be something that exists sort of in the bloodstream, where you can take multiple blood samples from the same person over time, and it's not particularly invasive. And an age-related biomarker also has to be something that works in a process that both humans and sort of the model organisms have. Otherwise, we won't be able to use model organisms to kind of study these biomarkers further. And that can slow the pace of research down a lot, right? And so an age-related biomarker needs to be involved in aging, able to be tested pretty easily without harming the person, and also needs to be something conserved between humans and lab animals. And some examples of biomarkers that are already being used for different processes are over here. And so I think one that's sort of a little bit easier to understand is using a biomarker to diagnose a disease. And so if you can identify a specific protein in fluid of patients that comes from their spine who have dementia, you can then diagnose them with a particular neurodegenerative disease. So in this case, the biomarker is a protein or proteins A beta 42 and tau, and they're something that is able to be tested repeatedly by removing spinal fluid from patients, and then you can diagnose Alzheimer's disease as the type of dementia that these patients have. You can also use biomarkers to monitor status of diseases, and then what we're talking about in terms of systems biology and aging to be predictive, right? And so these are just some examples of how biomarkers are already being used right now. But in order to identify the age-related biomarkers, we still have to do some more studies. And the one type of study that we do on people related to aging is what's known as a cross-sectional study. And so a cross-sectional study is basically a study that compares the mean or the average rate of change between two groups or two cohorts of people at a single point in time, right? And so you look at one specific day, two different groups of people, potentially of different ages, and then you compare the mean difference between those two groups. And a cohort is a group of individuals of the same age, but more importantly, they're also of a similar life experience. And so at this point, we know how important environmental factors are to aging, and so that's taken into consideration when setting up these cross-sectional studies and choosing your cohorts. And what's nice about a cross-sectional study is it's relatively easy to do and it's relatively cheap, right? It's a one-day thing. It's probably questionnaires or one-time measurements, and then it's a comparison between two big groups. And they can help provide some general knowledge in terms of aging, but it's not, cross-sectional studies are not specific to understanding sort of individual rates of aging. And so what you can see here in this cross-sectional study of mice on the right-hand side is really just looking at comparing the body weight of younger mice at 90 days of age and older mice at 600 days of age. And so you can make the general conclusion that body mass increases as age increases, but what it doesn't take in, but this study does not take into account is the fact that you have some individuals who are 600 days of age that have almost the same body mass as ones much younger. And you also have some very far away from the mean that are much larger than even those that fall along this mean line for weight at 600 days of age. And so this variation increases with age, and that's likely due to those environmental factors. And so that's one thing that cross-sectional studies don't necessarily take into account, is that as animals or humans age, there becomes this large spread or variation in phenotypes. And looking at the means and comparing them is not always necessarily the best thing to do, especially if you're interested in identifying things that are specific to an individual. And so we've also tried to study age using what's known as a longitudinal study. And so originally a longitudinal study was thought to be a way that we were going to be able to make some conclusions about the rate of aging in an individual by basically studying the same observation of the same characteristic in the same individual over a long period of time. And so rather than a cross-sectional study where we just look on one day and compare two groups of individuals of different age, this time, longitudinal study, we're going to take the same person and repeat an observation of the same characteristic. So in this case, the graph here, grip strength, in the same person over time, right? So over 100 years. And so these do provide some information, but it's also, again, tending to not be, it has a wide spread between individuals. And so longitudinal studies do provide valuable information, but more about how populations of similar people age rather than how individuals age. And that's, but they are more accurate in terms of reflecting how aging happens. And as you can see here, that there's sort of this gradual decline, not a dramatic linear drop-off. And that is accurate to how we know that aging happens, right? So that there's the sort of period of optimal function and then that gradual decline with age. And so they are useful for certain things. And one way that, we're really hoping to push the identification of biomarkers forward is what's called the Precision Medicine Initiative. And this was a pretty recent initiative by the Obama administration about 5 years ago. And its main goal was to start tailoring the prevention, diagnosis, and treatment of disease to the individual rather than to a large group. And so right now, the way that we treat people is basically by conducting a study and then creating sort of a representative treatment that we think should work for everyone. And the idea of precision medicine is to actually tailor every individual treatment, prevention, and diagnosis to you as a patient. And so the one way we can do that is by identifying biomarkers in ourselves, and then we'll know how best to treat and prevent diseases in the future. And this initiative is a combination of work from the National Academy of Sciences, the NIH, and the FDA. And the idea was to create a gigantic cohort of people, 1 million people to be exact, who can contribute their biological information, so DNA, and other different biological markers, as well as environmental data, so personal information, to create this open access database with a cohort of 1 million people, which systems biologists and other types of biologists can then go into to start studying and making some conclusions and potentially identifying biomarkers related to age, as well as to other diseases like cancer, type 2 diabetes, and things like that. And they've actually made some progress using this sort of precision medicine idea in treating cancers. And so you can look at cancer-related biomarkers in a specific group of people and identify the best treatment method for those based on their own personal medical information. And the ultimate idea behind precision medicine is to get the patient more involved and back being an active participant in their medical care, because for a while, physicians were the ones who had all the information. And so doctors were telling you what to do based on statistics of what worked best for just like a general group of people. And now with precision medicine, you have your own information, the doctor has the information as well, and you guys can work together to sort of choose the best options for treatment and prevention, and tailor them to yourself rather than to what worked best for the group that they tested those treatments on. This precision medicine initiative is still going on, and they're continuing to find biomarkers related not only to age, but also to diseases. And it's really cool, so I encourage you to look it up and take some time delving through that information. And there's also more about it in your book if you're interested.